So we've reached the last session of this workshop, and for this last session I'm happy to announce Aleksandar Miladinović, who will talk about time frequency analysis and how it can be used to better understand the cortical control of gait. After the first theoretical part, an interactive hands-on session will follow, so you can also download the materials that were sent to you via email in the morning. And after the talk, Aleksandar will also join us here to ask any questions. Enjoy the talk.
So let's get started. So in this lecture, I'm going to give you an overview of time frequency neural time series analysis, and I put in the parentheses gait because this is one topic that I will tackle during the lecture. But the concept of this lecture can be applied also for other experiments, and the presentation is going to be more generic, and I think it might be also useful for people who comes from other disciplines. Of course, it's a brief overview of time frequency analysis, especially power analysis, and what are advantages and what are limitations of power analysis, and we will also talk about certain aspects that are not always present in the broad literature, and also discuss and pose some open questions on the topic of the spectral analysis. So before starting with the lecture, I would like to give you a short outline of this presentation of this lecture. And the plan that initially I introduced the spectral analysis, and I introduced it on the more traditional way that we are used to do spectral analysis, which is called spectrum analysis. Then I will jump to the dynamic spectrum analysis, which also incorporates time axis and helps us to understand the progression of a certain phenomenon. And then we will also go to the very, very important aspect of the spectral normalization. And of course, as this is a workshop, I would like to show you some demos in the MATLAB and provide the code and the data so you can try yourself. We already heard about the time-based analysis, and in particular, event-related potential analysis in this workshop. And here I'm going to give a brief overview about what's the difference between the time-based analysis and the spectral analysis. And in particular, the first point is very important, that time-based analysis, since it's based on the average of the large amount of trials, is not suited for non-phase locked signals. What does that mean in practice? So that means that if you give a freedom to participant to start a certain task, and you are out of control of that, you actually don't know where the subject started. So this can introduce a various time onset from the stimulus presentation, let's say, which actually will completely obliterate the signals at the end, because this will cancel each other. Where does the ERP are suited? Well, mostly the ERP are used for in the cases where there is no voluntary control and voluntary control of the certain actions, such as visually evoked potentials, or even like P300 potential that are erased by a certain stimulus that draws attention to a subject. So it's subconscious, and we are out of control of it. Whereas when we ask subject to perform a certain task, for example, motor imager to move the limbs, from the point where we give instructions and the subject performs certain movement, the time can vary, and then in this case the time-based analysis is not usually a good solution. So you can see this effect, for example, that we have a multiple trials here, okay, and in this particular case, it's chosen to subject closed eyes, even though this is not a particular task related to the certain stimulus. It actually causes an increase of the alpha rhythm. If you would try to perform on such task, simple task of closing the eyes, the earpiece, you will end up with basically nothing. But that nothing is actually not really true. It's just nothing because these signals, these trials, are not phase locked. And then if you do a power analysis, for example, you calculate the alpha power, then you can see that actually and there is actually information in that. And then taking multiple trials will result in a shape that looked like this. So this is another parameter that can help us to estimate. So it's very useful to utilize power in such cases. The second point why ERPs are not preferred is because there are many kinds of dynamics in the EEG. They do not have a representation in the ERP. Okay, so we have a very limited number of prototypical ERPs that we know to exist. Motor-related cortical potentials, P300, P200, etc. But this is a limited number, and many other activities in our brain are more related and more studied from the oscillatory perspective. And we arrived at the third point, where the neurophysiological mechanisms that produce ERP are less well understood compared to the neurophysiological mechanisms that produce oscillation. Because with the advances of the technologies, we actually could arrive to even the single neuron firing, and that can help us to explain maybe certain oscillation that are measurable even with EEG. While with ERP, the exact motivation for the synchronization that give rise to, for example, a P300, it's not yet well understood. Okay, then to give a more wider introduction to the time frequency based approaches, there are three major advantages of analyzing and interpreting EEG data using time frequency based approach. So as I told you, it can be interpreted in the terms of neurophysiological mechanisms of neural oscillation. So if you can actually go from like measuring the simultaneous activation of multiple neurons to the single activation of a single neuron in certain cases. So this is actually the second point, where the actual neural oscillation is studied using highly biophysically detailed computational models in vitro, single cell recordings in vitro, microelectrodes in non-human animals, and then of course intracranial EEG in human patients. Furthermore, also EEG captures a dynamic and multi-dimensional space of brain processing. Then the ERP reveal a fraction of that space, and time frequency based reveal more. So what this want to say, that mostly the ERPs that we can see are a product of the low level oscillation that is certainly a single part of the whole neuronal activity in the brain. Well, the time frequency analysis, we can have a wider range of, let's say, another dimension that we can include in order to study certain phenomena. Okay, so what are some of the limitations? One of the limitation is decrease of temporal precision from time frequency decomposition, and so why is that so? But let's say when we have a time-based signal, time-based analysis such as ERP, we can preserve all the time points, and therefore we can have a very high temporal precision. Just to remind you that EEG has a very high temporal resolution. So what happens when you have to estimate the power? Because what do? We do all the approaches, different approaches, including the Fourier transform, as actually an estimate of power. And in order to estimate with a certain precision, you have to sacrifice the time precision. So to increase the spectral precision, you have to sacrifice the temporal precision, because you need enough of time points, enough of data to draw certain conclusions about that the oscillatory properties of that signal, and therefore we sacrifice to the certain level, the certain amount, the temporal precision. And then there's the second very important point, and that's a danger of the paralysis of analysis, meaning that increases the effect increases because the researchers get stuck constantly performing new analysis with the different parameters while losing the sight of the real purpose of the study. So now let's start with the time frequency analysis, and as I already told you, we will start with a static spectrum analysis, and this is maybe not the proper representation, but properly written. I mean, because here I cross the time, because the time in the static spectrum analysis is completely abstracted and it's non-existent, and that's kind of nice to start with that, because this is the basis of the time frequency analysis that we will tackle in the next slides and later. And if you understand the static spectrum analysis and what are the problems of the static spectrum analysis and limitations, we can go easily to time frequency analysis as the extension of what we learned in this part of the lecture. So another slide that help you to understand why the frequency-based analysis is more advantageous in comparison to the time-based analysis in particular cases. And as you can see in the first graph, we have a sine wave to which we superimpose some white noise, which has the very broad frequency, very broad bandwidth. But still, for us, if just looking at this signal, it's not that easy to understand that it's a sine signal. But if you do a frequency decomposition, you can see that there is a prominent 3 Hz component here, and which can actually which is much above the noise. And the same thing is happening here at something like 5 Hz. And also we have here, this is even more particular case where we have 2 frequencies superimposed together, which gives us something like this, which is hardly understandable just looking from the traces in the time domain. So just a brief introduction and an overview on Fourier transform. So I'm not going to go into the mathematical and formal definition of the Fourier transform, just more intuitive idea. I would try to give some more intuitive idea by just looking at this graph. So the idea of the Fourier was actually that any time signal with in a certain bandwidth can be represented as a sum of waves with a different frequency and amplitude, so he came up with a certain formula that helps us to decompose such time signal into a series of the sine waves with a certain coefficients that we can later use to calculate the power for the specific components. So of course, the transformation works under a certain condition. And one of the conditions of Fourier transform is the stationarity. And even here, he says that any stationary continuous process can be expectedly described by infinite sum of sinusoids of different amplitudes and phases. And that is particularly true, because what actually Fourier transform is an estimate of certain these coefficients, and it expects that the signal is periodic, so it goes infinitely in this direction and in the other direction in the same manner without changing, and that refers to the stationarity. So now we come up with this important concept of the stationarity, and this is particularly important for us who wants to deal with the time series, the neural time series, and the idea of science of we try to produce replicable results, and this actually non-stationarity creates a lot of doubts in the analysis performed so far and the papers published in the scientific literature, and that's because the results of the Fourier transform are easily interpretable only for stationary signals. So as as I mentioned, the concept of stationarity is important, but let's go further and understand what this means. We might read some papers and even on the topic that criticize applications of the power spectral analysis, and they usually mention stationary and non-stationary signals, and then they if you go to the search what in statistical terms, they also introduce wide stand stationary signals and stationary signals, which creates a lot of confusions. And I just would like to try to provide some of definitions and some of the explanation of what the stationary actually means in our case. And then here I took two definitions from a book of Mike Cohen, and then he says that stationary is as a signal is stationary when its statistical characteristics do not significantly change over time. And on the other side says that a non-stationary signal is non-stationary when stationary is violated. Which is, these two definitions, I mean, also brought by the author of the book, are not very clear, and certainly it does speak about statistical characteristics. And let's see what are those statistical characteristics, and what kind of descriptive statistics we can explore to figure out whether the signal is stationary and non-stationary. So let's suppose that we have some signal. This is some noise, some white noise, to be more precise. And here I created some time window. And so let's suppose that I calculate the mean of the signal here, and I do the same, and I calculate the mean in the time window 2. And then I go further and I calculate the mean in the third window. And if these three means in the three windows are the same or very close to each other, that means that the signal is mean stationary. So let's see in the definition here that the estimate of statistic is independent of size and location of the time window windows that signal is mean stationary. So that means that it's independent of the size and it's independent of the location. If I take a broader window here, the mean won't change with respect to the mean 2 and to the mean computed in the window 2 and to the mean computed in the window 3. Okay, so this means the mean, it refers to the mean stationarity. So let's suppose I take some other signal, and some other signal that's more closer to the EEG signal, and we can try to do the same operation. So I calculate the mean in the time window 1, then I calculate the mean in time window 2, and then I calculate the time window the mean in the mean of the signal in time window 3. And now my question is, do these three means differ? And the answer is yes. I mean, you can clearly see that the mean in this window is much above from the mean in this window 2, and it's probably also different from the window 3. So now the question is this signal or the part of the signal that mean stationary? No, it isn't. No, it certainly isn't. And so it's very close to the EEG signal, and this poses an open for discussion whether certain analyses are applicable in this case. But let's go further and explore some other non-stationary or stationarities. And one of the non-stationarities is so-called frequency stationary, but amplitude non-stationary. And here is, of course, this is not a real thing. This is artificially generated sign that progressively increase the amplitude, and that means that the amplitude is non-stationary. And the amplitude non-stationary, it also produces an effect that is hardly explainable by the Fourier transform. So this effect you can almost relate to for example, the burst of alpha in the certain conditions under certain conditions in the subject, that the frequency stays the same, but the amplitude changes over time. On the other hand, we have a frequency non-stationary, but the amplitude stationary. So the amplitude stays the same, but the sudden frequency changes. This is very rare. I don't know. It certainly exists also in EEG. So now the really important question is EEG stationary? And the answer to that, so now we come to the big question about the EEG and its stationarity. And the question is EEG stationary process? And the answer given in the paper called Everything You Wanted to Ask About EEG, but you were afraid to get the right answer. And the answer number 4 of that paper is says that no, EEG signal is non-stationary. And in general, biosignals are 3N, which stands for non-stationary, non-linear, and noisy. And here it's another, this is the definition that I already mentioned, that the non-stationarity means that the signal statistical characteristics change with time. And that the brain is actively essentially non-stationary. And now, let's say another very, very important question: can we use Fourier transform? And the answer is yes, because yes, because the Fourier transform is a valid operation. It will provide results, so we can get the coefficients even for non-stationary signals, because it's a valid mathematical transformation. Just wanted to tell you that so nonetheless that the transformation will work. The results are easily interpretable only for stationary signals. Okay. Now your question is how we can deal with that. What we can do to either make EEG stationary, which is, we don't want to do that, or how we can and where we can apply then the Fourier transform, under which conditions, and what are some things that we can do to let's say get rid or do something with the stationarity. And just to demonstrate you that the Fourier transform, in this particular case, and the implementation of the fast Fourier transform is applicable to the non-stationary signal that we actually produce, that we actually already checked that it's at least mean non-stationary, at least. So we shouldn't use the FFT, but nonetheless it will give us the results, and that results, if you look at this in the frequency domain, you could actually see that the shape, expected shape of the EEG signal. We have the high power and the low frequency, and then we have some peaks that might be alpha, and it's very, very similar to what we obtain with EEG. So should we do this? Should we give the whole signal, or even the whole and the whole length of the EEG, and put it directly into FFT? Well, we shouldn't do that in general, because this actually we can run into problems where the estimated spectra is actually not valid due to the non-stationarity that we mentioned before. So what we can do, there is another approach that tries to deal with this, and this is called Welch method. And the Welch method, also called Welch periodogram, is a method named after Peter D. Welch, and it estimates power spectra by dividing signal into successive blocks, forming that periodogram for each block and averaging their spectra. So let's say instead of having everything in the putting the whole signal in the FFT, we actually start by dividing our EEG, or our neural time series, into smaller windows, and then calculating the spectra. So as you can see that in this window, we assume that the signal is stationary. So in these small chunks of the data. So what we go, we then move this window, and then we calculate the spectra for all windows that you can see, and then we can calculate the spectra, the average spectra of the signal. So you can see that this window overlap a little bit, and I will tell you why is that, and why we didn't just put the windows next to each other. I will tell you more about that later, but just before that, I wanted to show you the differences between the FFT power spectral density estimate in comparison to the Welch power spectral density estimate. So as you can see, this is an example that that can be different, and yeah, so we actually get different results. Now let's go back and analyze the time frequency, the windows and the overlap. So the first of all, the overlap is desirable in this case, and it should be always used. And that's because let's suppose that if we have some signal here, of course it's a simple sine wave, but let's suppose this is an hour signal, and if we just use a window and we cut the signal in this particular two locations, what will happen here, and we apply the FFT in this particular window, the edges that I cut here will actually be misinterpreted by the FFT as a high frequency component. So the very sharp changes of the EG signal in these points, which will introduce the high frequency component that are not actually coming from a signal. So what you can do in this particular case, you can introduce tapers, so-called tapers, or particular windows that they usually have a Gaussian shape, and that starts from 0 and then rises and goes back. And application of such windows actually helps us to completely attenuate the beginning and the end of the signals. So when we apply the Fourier transform, the fast Fourier transform on this segment, there are no artificially induced noise by the sharp edges if we cut with the normal window. And so what are implications of that? I mean, what about the signal that we suppress that exists here and on one and the other side of this so-called taper? Well, there is nothing wrong with that signal. It's just that we had to do this way in order to avoid some errors in the frequency estimation of our chunk. So what we can do, we actually apply the overlap of windows. So the next Gaussian, next hand window, so-called, will start here, and then it will rise, and then it will cover this part of signal that's been lost, and so on. So we can go further with the overlapping windows and consider also the part of the spectrum that was not considered with the previous window. Okay. So, and also to be clear, there are many different type of windows. Then the hand window is most commonly used window for EG. So let's see now the comparison of the FFT and the Welch method, both very commonly used in the EEG time series analysis. And but we have to be really cautious when we read a paper where they apply the so-called full FFT method in their analysis. And also we have to be careful. Not everybody actually calls this approach Welch approach. And some people prefer to explain the procedure, how they obtain their spectra. So that might be very similar to the Welch approach. And also I'm just wanted to tell you that these two are not the only existing approaches to estimate power in the neural time series, but they are of course the most commonly used. And that's why I decided to include in this presentation. So let's dig in a little bit more about this comparison. And what is important to know is that the full FFT, what we see and discussed previously, is sensitive to noise. So the appearance of the burst with a certain frequency in the signal, even for very brief periods of time, or even at once, can actually lead to overestimation of the power in that frequency. And also, if we have the Welch approach, in the Welch approach, the burst will actually appear only in one or in a very limited number of windows. And then if you average a larger number of windows to that small number of windows, you actually will cancel out and you will reduce the influence of that noise. This brings us to the second question. So as we gradually, in terms of the in the time axis estimated the spectra, we then on the spectrum we actually are become less sensitive to non-stationarity in a Welch approach in comparison to the full FFT. So here it says robust to some non-stationarity, and in particular the means non-stationarity. Now the last question, where the full FFT is really better and we cannot do anything about it, is the higher spectral resolution in comparison to the Welch method. And this is because both methods are actually methods that estimate the power or the power in a certain frequencies, at a certain frequencies, and in both of the cases you will obtain the frequency, so-called frequency bands with a certain width. So that means that the spectral resolution can be different for the same signal with a Welch and full FFT. And why is that so? Well, as I said, if you go and look at this, the definition of the fast Fourier transform, you will see that the spectral resolution of the full Fourier transform depends on the number of the points in the temporal domain you input in the time or in the signal that you put inside this function. And this is surely true, and but we also have to be careful with that definition, because we can artificially oversample our signal, and it's a question whether that will bring us more spectral resolution. So no, that won't bring more spectral resolution, because of course artificial will have a more high spectra, but higher spectral, let's better call it precision, will remain the same. So, but let's go back and let's say consider that we have a longer time for a full Fourier transform, and because in Welch method we have a limited number of we have more windows with a short period. Then we have less points in this case to estimate each of the spectra. So less points means lower spectral domain. So while this can have a spectral resolution, for example, 0.5 Hertz, the Welch method in some cases can have a spectral resolution of let's say 2 Hertz. That means that in this case you might distinguish the oscillation at 1 and 2 from the oscillation on 2 Hertz, where here these two will be actually seen as the same. Okay. And now let's go to see what are the particular application of static spectra estimate. And I have to tell you that we are mostly biased also for historical reasons, and especially in the clinical domain, that we refer of the changes of the spectra in the so-called and so known EEG spectral bands, which correspond to the traditional analysis, time-based analysis of performed in clinical settings, by saying that this signal is predominantly there is a predominant alpha, there is predominant beta. You can see that by looking at the traces, and influenced by that, we then started to quantify the amount of alpha and the amount of theta and for each frequency bands. And then a lot of papers still uses this terminology, which is completely fine, and there is nothing wrong with that. But as I said, we have to be careful when such analysis is applicable, and how this analysis is performed. So one of the application of static spectra is actually for more static experiments, how to sort of say. So one of such thing is resting state EEG. Okay, well, where you record I don't know, 10 minutes of EEG, and then you assume that even there is an intrinsic non-stationarity in the signal in that period of time, there will actually, those changes will in the mean will stay the same. Okay, so then even if it changes, you have enough data, and you perform preferably of course the Welch method, and you obtain the PSD, the power spectral density for that, and then you can integrate that and produce the power in the EEG bands. Okay, so it's completely, this is fine if it's done correctly, and there is nothing wrong with that. So to be clear, but what is more important, the part that this workshop is to teach you how to read certain papers, and what to pay attention to when reading some papers, and interpretation and discussion of and results of this publication is and the second point is how, if we would like to do a certain study and me at home, maybe somebody would try to perform a certain analysis. And if you use MATLAB and you're familiar with MATLAB, there is already existing function that's called pvouch. And I think that this presentation actually helped you to understand what the data, what the input parameters of such functions are, and what is the x. Well, that's self-explanatory, our data. But what are the other two parameters, and how to set these two parameters to obtain nice and interpretable results. Okay. So let's now start talking about the time frequency analysis and so-called dynamic spectrum analysis, where the time is not crossed. And we talked about the definition of non-stationarity before and its relation to the time. And you might have an impression so far that this non-stationarity is undesirable, but actually that's not completely true, because as we could see in one of the explanation of the biosignals, and in particular signals that comes from the brain, and they are intrinsically non-stationary. The signals are non-stationary that comes from such sources. So getting rid of this non-stationarity is not actually what we want. We want to study these changes of the spectrum, these changes over time, and in particular these changes for our purposes and for example for gate analysis. I mean, there is no sense if I would consider and I would completely abstract the time domain of this this task, because then I might end with wrong results. I mean, not wrong results, but not interpretable results. So before going into what dynamic spectral analysis is, I would like to show you just briefly what I'm talking about. Because before we had a two dimensions, so a single line drawn that tells us what is the power spectral density of our signal, but we have to now introduce another dimension, and another dimension is time. So what is the scope of this part of the lecture that actually refers to dynamic spectral analysis is how to create such time frequency plots that on one axis it has a frequencies, and on the other axis it has a time. And this is not from the gate experiment, but from some other experiment. And in particular, we will learn how to produce such plots. And I also will provide a MATLAB code so you can try to do it by yourself. And but we won't stop only just creating 2D plots. We want to make use of certain tools that helps us to not only to visualize such plots, because usually these plots are made on a single electrode or electrode cluster, but we want to explore where these sources, or let's say signals, come from, and we can do that with the help of a certain tools that I will give you, and also give you a link to where you can download. Those softwares are not built by me, but I'm bringing them here because they're very useful in our case. And in particular for our gate analysis, if not for at this stage for study certain phenomena, but at least as a sanity check that these signals and this modulation that you can see here comes from the brain sources that are from brain centers that are related to the gate. Okay, so as you can see here, we don't have time, but we have the gate cycle, so that goes from 0 to 100, and we will in detail talk about that in our demo, because the gate cycle and the length of the gate cycle can change for a person, and that's very, very, very important to consider. Okay, so now let's go further, and we can question ourselves, okay, how can I create such colorful plot? Okay, And one of the simplest answer, well, we can use so-called short time Fourier transform. Okay. So we actually figured out that what the Fourier transform does, and then the short time Fourier transform is an extension of the Fourier transform that addresses the main limitation, the abstraction of a time. So how it works? Well, instead of actually putting the whole thing, we do something very similar to the Welch method, but we actually don't average those time windows. Instead, we calculate the spectra in a small frequency in the small time windows, and this is like how it looks in the frequency domains, and then we use that particular instance in our time axis. So I can see this thing here correspond to this the graphic here, and then we can do that for the whole time axis. Okay. So what I wrote here is that we can use FFT to extract the frequency structure of brief segments of time windows, rather than the entire time series. Okay, so instead of the whole time series, we take a brief signals and then we plot that. And of course, we have to use tapers for obvious reasons that I already explained. And this approach of the short time Fourier transform is completely valid, and there is nothing wrong with this process. But let's say it has a certain drawbacks, and these drawbacks of the short time Fourier transform are related to the time and the frequency windows that are always fixed across all frequencies in the frequency axis, and also in the time axis, as you can see. And if we go back to the idea that including FFT and also wavelets are the approaches to estimate spectra, and that our spectral resolution, how well they estimate, depends on the data the length of the data, of the number of the points you input there, you come to the conclusion that the number of cycles, okay, in the lower frequency bands span with a wider time window with respect with the higher frequency. What that means? That means that I can use a smaller time windows with a higher frequency, because I have enough point to obtain the frequency resolution, and the higher wider windows in the lower frequency, because here I might put like a single oscillation in this time window. In this time window I can fit the oscillation in half, and so on and so forth. So I can fit here more than a single oscillation in these frequency bands. And by reducing the time window, I basically, in the higher frequency, I drastically increase the time resolution of the frequency of the time frequency analysis. And if you remember at the beginning of this talk, I talked about one of the limitations of the time frequency analysis, as that we have a reduced time precision, and that is true. But we can clearly see that this time frequency, we can come up to some compromise using wavelets in relation to the standard short time Fourier transform. So why then we still use Fourier transform, and can we obtain the similar results to wavelets? And the answer to these questions are that we use short Fourier transform because of the historical reasons, and there are particular applications when they are useful. But what is more important, can we get similar results as we obtained with the wavelet? And the answer is yes, but this procedure, creating a different time windows for a different frequency, is more complex. And this gives us some more information about the Morlet wavelet, which is a particular type of wavelet, because the wavelet, there are multiple types of wavelet, and wavelet is actually the diminutive of the wave, so the small wave wavelet that has a particular had to satisfy certain conditions. So this Morlet wavelet is nothing more than a sine signal tapered with a tapered with a Gaussian kernel, which is the red thing. And as the result of that, we got the black line, black signal that's called wavelet. And the two particular information which are not important, you can look more on the videos about the wavelet design, is that it's how it's centered, that the kernel goes to 0 at the beginning, and at the end, at the area of this wavelet under this curve as unitary. Okay. So now we learned how to create a wavelet. We said that you need a time window, that you need a sine signal, that you need a kernel, that you apply that you multiply these two, and then you create a wavelet. Okay. And then a question, what we do next? And of course, we create multiple wavelets with multiple frequencies. And then what we do is we applied convolution. And convolution is a specific operation in the time domain that will help us to produce the power by using that wavelet and the time signals. And it basically overlaps the signals and looks where this overlap is maximal. So if we create a wavelet of the size 6 frequency with the 6 Hz, this 6 Hz is actually overlapped with our signal time by time, and then we calculate this similarity, this overlap of our signal with that particular 6-band frequency. This will give us the real value representation on amplitude, and by squaring that amplitude, we will get the power of that signal. Of course, this wavelet, I just showed you the representation before and how to produce it, but this refers also only for the real part of the wavelet. The Morlet wavelet, and a particularly complex Morlet wavelet, is of our interest because they help us not only to estimate the real part, but also to amplify the phase of our signal. And this is how the Morlet wavelet looks like in the real, in the imaginary and the real axis. Okay, and this is how it looks with the full line. It's the real part and the dotted line, the imaginary part. Here you can see different wavelets created with a different frequency and with the different Gaussian kernel. Okay. So this is topic related how the spectral power and the shape of the Gaussian are the spectral resolution and the width of the Gaussian kernel are related, and how they can produce a different time frequency plots. So in the literature, usually you would see the definitions of the wavelet, and when you want to create a wavelet, you have to input the number of cycles of that wavelet. Okay. And clearly that in the particular time frequency plot, the 3-cycle wavelet, which means a very, very narrow Gaussian kernel, gives us the very high spectral resolution, very high temporal resolution, because it will capture the changes for very, very brief time windows, so to say. But of course, this problem, the other spectral resolution on the other side, will deteriorate drastically. And this is the same data plotted with a 10 cycle, so more wider wavelet, more wider kernel. The 10 cycle actually refers how many of the sine periods remain inside below the kernel after tapering. And what you can actually see here is that here, where the temporal resolution was poor, I could not differentiate these two blobs, because here I can see two blobs, whereas here it looks like a single. But if you look at the time domain from the time axis, I clearly see that here something that was very, very different, now it became a single blob. What is usually done today, and what we are going to do, is so-called variable 3 to 10 cycles, and then you take the mean of these plots from 3 to 10. This is usually the number of the cycles you want to do, and then you come up with a certain compromise of that. But this is another very important parameter that you have to consider when interpreting the results, and how the results can differ if you use different input parameters. So to read about this, how to define wavelets, you can find a paper from Michael Cohen. It's called A Better Way to Define and Describe Moderate Wavelets for Time Frequency Analysis. And he actually explains there what are the good way and how we should use the time moderate wavelets for EEG analysis. Now, I just wanted to tell you that the wavelets are the preferred method for the estimation of the spectra. It's also as it's also available in the standard EEG package. If you click, for example, channel TF, channel time frequency, and then something like this will appear, and then now you learn what this wavelet cycles and minimum and maximum wavelets or sequence, he says. So you can input the sequence of different wavelets here to produce such time frequency maps. Or you can, as an alternative, you can use FFT. So this refers as a short time FFT. Okay, so you now learn one important part when you want to do the time frequency analysis, even in EEG lab. So what are the advantages and disadvantages of wavelets and short time FFTs? Well, the short time FFT requires more parameters, it's lower computation and lower temporal resolution, where the wavelets require fewer parameters, faster computation, and higher temporal resolution. And of course, the short time FFT in wavelets are not the only type of time frequency spectra estimation approaches. There are many others less common, such as Hilbert transform, such as multi-tapering FFT, and etc. So these are just the most commonly used, and I confirmed that by showing you the picture from EEG lab, and why you should. And I've just given an explanation why you should use wavelets and where you should look for particular for the parameters that can create ambiguous results and maybe introduce some problems. So now we go further on a very, very important topic, and this topic is regards the spectrum baseline normalization. And for those who are familiar with EEG spectral analysis, but also for those who don't know much about it, I might hear about something, or actually saw the previous images. The spectra of EEG actually follows the 1/f rule, and the 1/f rule tells us, and this is not only specific to EEG but to other biosignals, is that the signals with a lower frequency oscillation have higher power with respect to the signals of higher frequencies. Okay, and if you look at this time frequency plot, it tells you the very same thing. So the color here refers to the power, and the low means low, the red means high, and you could see that virtually if you go up and up to 45 Hertz, the difference is so prominent that even the slight changes that can happen here will be completely obliterated by the scale of the effect that happens in the, for example, at 10 Hertz. Now the question is how we can deal with that, how we can actually perform the EEG analysis and make such EEG plots. If our hypothesis tells us that there will be something happening at 45 Hertz, and we also want to measure the whole from 1 to, let's say, to 45 Hz. And how we can see this and how we can compare this effect. Let's say in an example we expected a 45 Hz, but we also want to measure something what's happening at 10 Hz at the same time. And so the answer to that, we have to use Bayesian normalization. So here I'm giving you a particular example how the time frequency plot can differ in the case of there is no baseline normalization, and in the case where the baseline normalization is applied. What you can see is that that certain features that are available in the lower frequency time frequency plot are not available in the upper frequency plot, which tells us the importance about the the time frequency the normalization in the time frequency analysis. And we can also see here that this time frequency plot is very affected by the existence of the alpha, and but this alpha is not related to our experiment. And if we normalize the existence of that alpha here, it will actually make this time frequency plot free of this alpha contamination, so-called, and it will reveal only the changes that are relative to the baseline that we have selected. It also gives us the possibility to analyze, for example, the data on the very low frequencies and also the data on the high frequencies. And if you take a look, for example, here, the blob here it's existing in the normalized time frequency plot. It's virtually non-existent in the upper time frequency plot because of the scale of the effect of what's happening there. So to say that the baseline normalization helps us to transform power data to the same scale. It helps us to normalize and put all power results into a common and easily numerical interpretable metric. It helps us to normalize power data. It helps us to make this normalized power data more applicable for a parametric statistical test later, because it usually shows it's often normally distributed. The first point is particularly important, especially if we want to do inter-subject comparison for a certain task, as the power itself can vary across the different individuals and can be affected by different factors such as the skull thickness, etc., etc. By this normalization, we basically can come to the unitless measures that can be expressed or in the decibels or in the percentage of the changes of the spectra relative to the baseline. And of course, the selection of baseline is not a trivial job. It has to be done. It has to be performed in a meaningful way because the selection of the baseline can lead to a different time frequency maps, and this can be easily seen in these three images how this differ because we changed change the baseline from this time window to time window 2. Okay. So how do we select baseline? This is not a trivial question, and should be also and always incorporated in our experimental design. In the experiments that are performed inside in the lab and are more, let's sort of say, traditional experiments, the baseline it's usually chosen to be some silent periods of the experiment, such as pause, and then when the condition of our experiment appears, we perform the time frequency analysis. But when we talked about the gate, this is not that easy, because first the baseline we don't have such easily be distinguishable conditions where we can use for the baseline. Of course, someone might say we can use standing as the baseline and then the person walks, which is one of the options, or we can use multiple baselines, which is even a better option. So if you have a person standing at the beginning of the beginning before it starts to walk, and then walks the distance that's designed by the experiment, and stops and then waits for a little bit and goes back, and those silent periods, the periods of standing, can be used to calculate the baseline. That's another option that also should be considered. Of course, if this option does not actually influence the behavior of the subject, which is not easily predictable and should be actually tested. And then the third option is that we don't use any of the previous, but we actually perform the mean, so we calculate the mean power in these time frequency windows, and then we apply the mean power normalization. So why the baseline at the beginning of the standing is not a good idea? Well, in one of my papers I showed that how these power features drifts in the resting state EEG in the subject that we recorded at the beginning of our one session, and then we waited 20 minutes with the cap on, and then we recorded on cam another 5 minutes. So what you could see, another 5 minutes of EEG, of course, and then you could see that there is the changes of the mean of the power features, and which tells you that there is a drift. There is a drift in the power features during period of time, which means that the baseline that we selected, for example, if standing and not corrected during the experiment, can be actually produce misleading results, and maybe should not be considered in the analysis. Now you can always in general have two different baseline normalizations. You can actually condition specific baseline. That means that each trial is normalized with its own specific baseline. This is not commonly used because usually you want to have a baseline that is clear of the artifacts, which is extremely hard with the EEG, and let's not talk about even walking EEG. So what usually people do is condition average baseline. Condition average baseline is that I take the baseline of multiple trials and then I average that baseline, and I use this baseline for normalizing the rest of the time frequency map. Once you select the baseline, and you can select it a little bit wider, and one of the indications that you selected a good baseline is that you don't see much changes in this baseline. So here the green color refers to zeros. There is no changes, and if this particular color is maintained in the selected time window for the baseline, you might actually have a certain conclusion that this baseline is well selected. Of course, you have to understand that there are also other statistical methods that you can use to help you to assess whether you select the good baseline, and you can check this out more in the lectures and in the book of the that you can find on this website. And this particular book is also written by Michael Cohen, and it's called Analyzing Real Time Series Data. And it goes credit to this book because many of the pictures and many of the concepts important for the time series, near time series analysis, are taken from this book. And in hands-on session, I will show you a brief presentation. Then we will pass to the MATLAB code. And here I just wanted to introduce certain aspects that we already discussed previously. And in order to perform the hands-on session, you would need an EEG lab. The EEG lab is the plugin very commonly used. You can download it from the link below. You will also need a Brainstorm plugin, and optionally, which is also optionally, you can check out the code from the Mike Cohen book, which is given below. However, the code for the time frequency viewer will be provided for this workshop. Let's talk about the data. The data used for this experiment is data acquired in the TwinBrain project in Trieste, Italy. The data comes from the participants who performed single task walking in the lab environment. During that time, we recorded motion data and EEG data at the same time, but also some other data. For this session, what is in particular, what would be for the particular use is the motion data and the EEG. What you can see here in the screenshot of the code, the first code refers to the inclusion of the path of our EEG lab, certain toolboxes, including our EEG lab. You can also add a path manually by right-clicking to the folder where your EEG lab is recorded and where your Brainstorm, and in particular time frequency toolbox of your Brainstorm is located. So right click to the folder, you can and then select add this folder to the sub to the path, and then you can omit these two paths. This line EG no GUI starts the EG and initialize the workspace with the needed variables for further analysis. The code is actually, certain parts of the codes are borrowed from Martin Sieber, who you can check his presentation also on the MoBI YouTube channel, and especially the parts that are referred to the cleaning of the data, so selection, bed IC, but channels. In this case, I didn't put any channel, but left it just for compatibility purposes. So if one want to reuse the script for other data, the thresholding, which tells us what the parts of the data is bad, the high pass filter, so this means 1 Hertz, the gate cycle tolerance, that means that from the data, from our mockup data, which is motion data, so it's not always perfect, we will extract the markers of the start of the gate, in particular the heel strikes, and if these markers are not always perfect, or the data is not always let's say easily interpretable, then we have to define also the tolerance, how our gate cycle, the length of the gate cycle is considered into the normal, let's say normal timing, and if this timing exceeds 20% of the total gate cycle time, we won't consider these periods of time, because this could refer to, for example, to the period where the person arrived at the end of the walking path and had to turn around and go back. So this turning around is not of interest for our analysis. Then there is a variable called to apply the surface Laplacian, and also to define our wavelet parameter. And here this wavelet parameter can be defined either a number of cycles, or in with a variable that's called full width half maximum. And that full width half maximum is actually referred to the Gaussian kernel. So the width of the Gaussian kernel. You can check it online more about what actually this refers to. Okay. And then what we do once we load the EG data, we load the mockup data, we remove other markers, because for our analysis we are not interested for the markers that comes from let's say during the experiment, such as button presses, etc. So what we are interested here is actually heel strike, because we are interested in the activation of the cortex, the brain cortex, during the gate cycle. And so here what I did, I selected actually the only channels that are relevant for me to perform the heel strike detection. What I then did is I actually resampled the signal to 256, and this is not completely necessary. Why I did resample the signal is that I could match actually the time axis. I can use the same time axis for the EEG and the mockup at the same time, and so I simplify this by oversampling the data. Okay, by oversampling the data, I could actually have a single time vector that I can use later for the analysis. So now this code snippet that I actually showed here is related to left heel strike markers. The first parameter that we need to set in this case is to set how much of the time at the beginning of our recording we want to ignore. So because usually when you perform the EEG and you put the cap, the subject has to walk to the starting position, and we are not considering that for our experiment. So I put here, in this particular case, it's 5 seconds. You can change that, and you should change, and it's based. You should change based on your experimental condition about the real situation when you look at the EEG. Now, if you look at the presentation on the first day of the workshop, you might know that, for example, the channel, it's called leftput.pw, refers to the pressure sensor that's located, the heel sensor that's located under the left heel, and it's only present in the left foot, so to say, left foot group of the sensors with this naming. So for us, this is the channel that we are going to look for the changes, and that helps us to detect the heel strikes. The heel strikes are detected by simple find peaks that is available in the MATLAB toolbox, and then once we select the transitions, so because the pressure sensors will have a characteristic let's say square shape signaling, not that ideal, but let's say we can compare it to that. We want to estimate transition sides, so the rising and the falling transition side. And for us, the rising and falling transition side are not of interest, because we are interested in the rising transition side. That's related to the pressure of the sensors once the heel strike happens. So to eliminate the other point, we define the differences between two markers of the first two markers, and then we eliminate 20 points. We had 20 points as a tolerance to that one. So any plateau, any square wave that exceeds this, that means that the subject stayed longer on the ground, then this number of points we exclude, and this is done by simple removing these markers. Then we actually add these markers into the mockup event variable, which is a standard variable of the EEG lab data structure, so to say. And here are the results. So you can see the left heel pressure, toe pressure, and this is I plotted also the coordinates, the y coordinates. So the y coordinates helps us to as a sanity check whether this heel strike is performed during the correct movement of the during the gait. And as you can see, it's almost always in the same position with regard to the y-axis, and that y-axis corresponds of the how person moves the legs from the floor to the in the upper direction. So here we have a heel strikes. And as you can see. We obtained the markers for the heel strikes. Now, once we perform that, of course, you could hear more about the gait cycle analysis in the first day of this workshop. Now we go to the pre-processing. What we need to do is we want to filter with a high pass filter, because we want to eliminate any drifting of the signal, so we can perform also sort of say thresholding of the data, which is done in this particular part of the code, and elimination of the bad components. We also performed I took only 19 channels, because to simplify this is an optional. You can also comment out this, but it's going to take a little bit more to estimate all the power for all the channels. So just as a sanity check to check how the bad segment detection works. You can see I added as an additional channel to our EEG channels, where the 1 corresponds to the good segment and 0 to the bad channel, to the bad segments. And you could see that here it actually selected the part of the signal that it's bad. But actually this threshold didn't eliminate any of the signal, but it just assigned the 1-0 vector that corresponds to each point that exists in the signal, and then it tells us later for analysis that we only has to consider the parts of the signal that's valid. In that sense, we don't have to lose the whole epoch, but we can still keep, for example, the n% of the epoch, and then perform only spectral analysis for this particular epoch in that time. Why I also showed you that there is an option to turn on the Laplacian surface, called surface Laplacian. And surface Laplacian is actually one of the spatial filters, a very, very simple non-data driven spatial filter that helps us with a source analysis. It actually tries to reverse the effect of the volume conduction, that a certain effect that actually happens below the skull has been recorded by multiple electrodes and has been seen as a single effect by the surface Laplacian. You can localize more activities that are happening just below the electrodes. You can see here, for example, the same effect measured by without and with surface Laplacian, and you can see that this component is it's the topographical map that is more wider and spread over the whole head. So this is very simple, and it actually consists of actually weighting and subtracting the channels from the surrounding electrodes from this particular channel, and so for each channel we subtract the weighted sum of the other channels. Okay, and in that sense we are trying to make, for example, this channel invariant to the measured activity from the neighboring channels. This particular part of the code does this job. You can also try to switch on and switch off the surface Laplacian by changing the Boolean variable apply surface Laplacian. Okay, furthermore, then the gait cycle, as the gait doesn't always have the fixed time. and it can vary from also in the same subject, we need to perform time frequency analysis and averaging, let's say that correspond to the same cycle, phases of the cycle. So for that, we defined a 0 to 100% cycle analysis, and then we actually performed a later time frequency analysis using the Morlet transform function from offered in the Brainstorm time frequency analysis plugin to calculate time frequency plot. We do that for all channels, and we save that in the let me see here. It's not, maybe it's visible, but there is a the variable that contains all the channels. Once we do that, we can start on the Java script, and we can plot the results. Furthermore, we can explore the hour time frequency plot by selecting either picking up the electrodes and also looking how this frequency map corresponds to the topographical map for this particular location in terms of the time and here time in sense of 9%. Here's the milliseconds because the frequency map is made for the time dimension here, but that I forgot to change. And then the frequency axis. Okay, so now we can go and check out the MATLAB demo. And in the folder that is provided by the workshop organizers, you can see that there are some data, and what's particularly important is two files, which is mockup set, and this is our data from the shadow motion, so our information about the heel strikes, where we can extract wherefrom we can extract the markers, and then our EG data. Okay, here I also included the EG plot wide, which is the standard EG plot library, just optimized for the widescreen, Laplacian function, and also the additional time frequency viewer. Okay, so to initiate this, we have to first run the first snippet, the first part of the code that will help us to clean our workspace and close other windows if they are open. Then, as I told you, we load our data. We load our we add to the path our toolboxes, and we also set up the file names where we can load data and set up certain parameters that we already seen. And one so the important parameters is the bed ICs, the bed components. Here they're selected randomly. I mean, from the talk that's that was done before my talk actually described how you can select these components more wisely. This is a simple thresholding method that helps you to eliminate the bad burst of the signals, so you don't to mark it as a bad. This is high pass settings for high pass filter. The tolerance of the gate cycle that we already discussed. Application of the surface Laplacian. You can change it to false and see what are the results. And full width half maximum, which refers to the wavelet parameters for time frequency plots. You can also play with that by changing this parameter to 1 or to 6, and you will see the effect of such changes. Then the second part, once we performed we executed the first part, we can go to the second part. That helps us to load the data and then select only relevant channels of the data. It also performs the oversampling of the mockup data, and then also creates a timeline, a single timeline vector for our analysis. So so now, as you can see, we have our EG data, okay, which is we have a number of channels, and then we have the data. There it is. We have 124 channels per 76,000 points. And if we look at, for example, the mockup function, okay, there it is. We have 3 channels because we selected only irrelevant, and we have the same number of points. So we have 3 per 76,800, which corresponds to the number of points we have in the EG. That's why I actually oversample the signal, so we have the same time vector, timestamps that refers to each point. Now we go to the definition of to find the heel strikes, And here, as I told you, we defined the 5 seconds to ignore at the beginning of the signal. That should be changed all for each subject that you perform analysis for, because that also depends on the real situation during the EEG recording. So now if we run this part, okay, it will perform the analysis. It will perform it will find the heel strikes, but at the same time it actually will include this heel strike, left heel strike markers. If I press normalize, it will normalize my channels and maybe increase some scale, so we can easily see how the data looks like. We can also see that the first 5 seconds are ignored and that the first markers are put the after 5 seconds. This is what I was talking before. We can take a look at the signal. So if we are not happy with what's been done, I can also manually eliminate certain markers that are not related to the proper beginning of the heel strike. Okay. So let's go further. Yeah, this we can also let me see here. Yes, so we perform the insertion of the markers, and now we go to the pre-processing. We perform the filtering of the data, and then we can eliminate the bad ICs. The bad ICs are defined in this vector, and I already showed you at the beginning that we selected from 1 to 12 as the ICs that are bad. And we also select only 19 channels which correspond to the standard 1020 the standard 1020 distribution of the electrodes on the head. And this is only to speed up. I mean, you can comment out this section if you run your code, and if you have a computer that's powerful enough that can done this in fast. I mean, it's not that it slows up, but yeah, here I just selected 19 because maybe it creates the better topographical maps, and I wanted to check each electrode. So you can play around with this. Then we perform thresholding. You can check these conditions, and so if I run this, okay, before actually looking at the results, we can look what actually this vector means. And if I execute this vector, it should actually give me, okay, so now the code is posted, so it should actually give me the number, the vector of zeros and ones that has the same length as the data, the data points. And for each data points, there will be a definition if that data points is a good and bad. And if I plot that channel, as I already explained you can see something like this, where the 1 corresponds to the channel that the data is good, and the 0 corresponds to bad part. And as you can see here, it selected that this part of the signal is bad and should not be considered for analysis. So you can scroll and check this out more, as you can see, more cautiously. So as you can see here we have a burst that could be maybe even eliminated with certain pre-processing, but here we decided not to. And you can see that clearly that this part of signal is omitted from analysis. The same goes here. Okay. So let's go further. What is important here is that it tells us what is the percentage of the data that is valid, and then we can see that in this case 84.22% of the data is valid. So now I have to press any key to continue this plotting because I put the pause here. So if I press pause space, yeah, then it printed out the vector. Okay, so this can eliminate. So if I go further, we define the gate cycles, and for each of the gate phase from 0 to 100%, okay, we perform time frequency analysis. So this time frequency analysis, so the time frequency plot are actually saved in this particular variable. And if I type size, okay, of throw channels, you can see that I have actually 20 channels that I pre-selected before, 25 frequency, which is with the frequency resolution of 2 Hz, which goes from 0 to 50 Hz, and then I have 101 point, which refers to the gate phase. And why it's 101 and why it's not 100? Because in MATLAB the data goes from 1, the indexes go from 1 to not from 0, which means that in total we have 100 points here. Okay. Now, once we created such structure, we can use and plot our results. And if I do this, this particular window will appear on the screen. And yeah, this particular window helps us to explore more and to do a certain sanity check. What you can see before starting to actually explore what happens here, we can actually look at the y and x axis, and on the y-axis we see the frequencies, and on the x-axis we can see the gate phase from 0 to 100%. Here we can select the desired electrode that we want to show the plot for. So for example, I can tell for phi-z, or I can tell for the pz, okay? And this is the different time frequency plots. So let's suppose that we are interested in cz, and we want to explore what actually happens here. And this particular time viewer helps us not only to localize this point, but also helps us to localize on time frequency in terms of the frequency and the time, but helps us also to localize where this activity comes from dominantly. And as you can see, this particular what's happening at 13 hertz comes from the CZ. And what the blue tells us that there is a decrease of the 2. So we have to look at the color limits here. Decrease of the 2 decibels with the in relation to the mean power of the whole time frequency map. So there is a fluctuation. We have the decrease of the activity at the 13 hertz at the level of CZ on the around, let's say, 9% of the gate cycle. You can see how this changes, okay, and how this moves, and so you can explore how this goes back and forth. And then we see the huge increase here. So if I click here, we would see the increase of this spectra here. The same you can do for higher frequencies. You can click there and observe how this changes. For example, here we have something at the F7, F3 electrode. Even though that we look at the plot of the CZ, the spreading of this activity comes from this frontal part of the skull is actually affecting also our time frequency map. But this can be a noise, this can be something else. But so this is why it is important that we not look only to this data, but also look to the topographical representation of the data. So what else we can do? You can see that there is a clear alternation. There is a clear alternation of the increase and decrease of power relative to the mean power of that whole time frequency map. And this is in line with what Martin Sieber actually showed us, that this particular pattern also was present in the data from Berlin, and yeah, this data is actually confirms that his study that he performed, and you can check it out in his lecture on in the previous workshop. So I mean, you can play around. This is the idea of the whole workshop. This is just a proof of concept. In order to understand and to better interpret this data, it would be also nice that I put above all phases of the gate, so we can understand what actually this means here from 30 to 50%. Does this like huge activity that's in the PZ has something more meaningful? Okay, so let's go back to the functions of this time frequency plot. I mean, you can for sure here change the color limits, and then you can see how that affects the whole image. I mean, it doesn't change it a lot. Doesn't change the data, but what actually changes is the limits of the color. So they become more or less prominent. If you put usually 3 here, because this is done in the decibels, that means that what's something in the blue, like here, refers to the -3 decibels, which tells you that this particular event, this particular bulb here, is 2 times lower than the mean EG power at the same frequency. Okay, so this is that's why the 3 is a nice number that you can use for time frequency map. You can also go and find what is the maximum, and we can see here's the maximum, and here is the minimum. Probably, yeah, it's somewhere here. So so why is that? I mean, we still have to discuss and more and brainstorm about the results, but taking in consideration that this is done, let's say ad hoc, and that we selected randomly, and not in some more smart manner to remove certain ICs. And just clicking the activity that we could see mostly in the most of the cases comes from the central CZ electrode, tells us that the activity the activity changes across this time frequency map are actually related to the walking and not to something else. Okay, there are other options that you could explore. You can put without no electrodes. You can add just dot. You can put some numbers, labels. You can change like more smooth or pixelized, as you can see the power spectra. You can then save this for your paper. And of course, you can choose to see some other electrode. This is, for example, C4 electrode, and C4 and C3 are also important electrodes because the C4 and these are the electrodes over the motor cortex. And as you can see also here, there are particular changes in the patterns of in the frequency bands. And yeah, I mean here, if you click, you can observe the activation, and this is the left foot. I expect the activation on the contralateral side of the skull. Okay. I think that's enough from me. I have to stop here. If you have any questions, feel free to ask me. And of course, thank you for your attention. Okay, I will check out the comments and also on the YouTube later. And then yeah, thank you again for your attention.
Thank you, Alex, for this awesome, really detailed and nicely structured presentation, and also for adding additional, for expanding our toolbox. Basically, we can always get back to the things that you presented and try it out ourselves. So yeah, thanks a lot. I'm really happy for having you here in person with me. So I want to touch upon movement-related beta desynchronization. So you explained a little bit about how it happens and that when is it usually observed. And I was thinking about, like, do you think this could be a marker that could potentially be signaling clinical improvements or also the deterioration of the motor-related capabilities in clinical populations? What are your thoughts on that?
Yeah, first of all, thank you for inviting me. It was really my pleasure to present my work and to contribute to the knowledge exchange between the groups. You mentioned beta desynchronization, and so these notions of the frequency band are some kind of legacy of the clinical use in the past of the EEG. And the whole idea of the time frequency analysis is that we kind of broader the idea that we don't usually have to divide our frequencies in certain bands to study certain phenomena. Because as you could see in the presentation, a certain time frequency window can have the blobs of different size. Of course, you know, the literature in the past actually refers to the changes, certain changes, especially when we talk about pathologies such as Parkinson's disease, and so-called slowing of the EEG, which means that moving of the spectral power in the lower frequency bands. So if you see the EEG of a resting state EEG of a Parkinsonian, you will see that it has abnormal low frequency power and lower high frequency power. What would be interesting is that the scientific literature also adopts more broader view and also perform time frequency analysis. So it would be nice if we could do even a simple task, such as, for example, in the lab, motor imagery, and see how the time frequency, normalized time frequency, changes. And there are some works on that topic, and we could see that, for example, Parkinson's disease were not able to produce. So there is no so-called beta rebound after the motor imagery. And the motor imagery is also related. So the motor imagery, for those who don't know, is actually the simulating of the motor movement. And what motor imagery does is produces very similar cortical activation. In the Parkinson's disease, what's called beta rebound, so the burst of beta after the imagining or after the movement can be used, potentially can be used to measure as a measure of improvement. Mm-hmm. For example, let's say that we apply this protocol for the rehabilitation strategies, and we have some metrics that measure how they improve, or better to say, how they maintain the state of a motor control, so whether they properly walk. We might use to quantify, for example, the changes in the beta band, let's say how the changes during the time, not only in the beta band, can be exploited to see whether certain strategy for rehabilitation is effective, or it is less effective in certain cases, and whether they improve or not.
So how far do you think we are from implementing this kind of markers into the rehabilitation processes?
I mean, we are not there yet, for sure. The biggest disadvantage is that until now we didn't have technology that we could actually synchronize multiple devices, and for many hospitals this is yet unknown how they, for example, we heard talk about the EMG synchronization with the EEG. Here we have the contemporary EEG movement, and we use the markers from the movement to perform epoching, so to select the EEG that are relevant. In the case of the clinical application of EEG, unfortunately, we still stayed at the level where we actually use mostly resting state EEG, without actually having this communication between multiple devices that can give us more useful data to study certain pathologies. So we have first to resolve maybe technical issues, so like implementation of such hardware, and then we can move to the study protocols. And the study protocols is that our, like the project as ours, gives an important input how to design them, and how maybe let's go back to the first question: how can we track, for example, changes? If we manage to give the clear answer how this protocol can serve, in the clinical purposes, this for sure has a great potential for the improvement of to implementation in the clinics.
Okay, so you mentioned spectral normalization, which seems to be really like the key, let's say, decision based on which the results of the spectral analysis based on which they could change, they could differ from each other. So how would you argue, like, which condition to take as a baseline, as a normalization of your spectral analysis, and what kind of questions can you then answer using a specific baseline correction?
Okay, so good question, but I'm afraid I cannot give like the correct answer, because there is no a correct answer in this case. So it is clear that spectral normalization is fundamental, especially if one wants to do also inter-subject comparison, because you get rid of the units, you actually talk about the changes, relative changes to the baseline, and that helps a lot to, for example, compare between subjects, between groups, etc., etc. So, but the question, but also the very important question is, how do we choose that baseline? And if you have, for example, some experiment in the lab, some standard cognitive experiment, one can say, well, the baseline is actually what precedes the experiment. So you have a pose, the person stands there, does nothing, and then you have a task, he performs the task, and someone would say that's the baseline. What about gait? I mean, this is more, it's get more complicated, it's not easy to say what is the baseline that we can use, what is the baseline EEG. that we can use to normalize the walking EEG. And there are a few options I mentioned. One is that you can use standing EEG, for example, before the walk, and then use the computed power in that period to normalize what happens later. But the problem of that is that if you have a lot of time between the recording of the standing EEG and your experiment, or your experiment simply lasts long, it takes like 20 minutes to perform, there will be changes, of course, in the EEG that so that the baseline becomes not ideal in that case. So and not related to the task the subject performed, because what we actually want to see when we perform baseline normalization is to make the signal of the task invariant to the other neural oscillations that are happening and that are not irrelevant to what we are studying. Okay, but if we have a standing EEG and then after 20 minutes we have a walking EEG, there is a lot of changes already happening in the brain that this baseline cannot make our signal irrelevant. So I thought about this, and then how we can resolve this question, especially in the gait EEG. And one of the ideas is actually to perform a small pauses. So, for example, if you have a task to walk the certain distance, so at the beginning you record the EEG, you mark it as a baseline EEG, okay, and then you start walking. When you reach the other end, you stop for a while. It doesn't have to be so long. It's enough, for example, 2 seconds, 1 second. So you can use the baseline of the standing and that baseline to see if there is a drastic drift, okay? So you can correct it. But this input has to be implemented in the experimental design. So we cannot do that afterward, okay? That's the problem. So in the data that we have, there is no stop at the beginning, at the end. So as a compromise, I had to perform, for example, I had to take the mean power during the gait cycle, and then to show the time frequency map, how the data changes around that mean. Okay, so this is a compromise. But I assure you that if we would have also the baseline periods, okay, the time frequency map would be even better, and it would even better convey the activation of the motor cortex related to the task.
So definitely something to keep in mind when conducting the next experiment. Okay, well, my next question would be if we think about the frequency spectra, right? Yeah. So some higher frequencies are also modulated with respect to gait. So there's a gait-related cortical modulation, but I assume and I think that also this activity is superimposed on the muscle-related spectral changes that occur because of the gait itself. Mm-hmm. So what would you say is the best approach of separating the two to really get to the cortical-related gait spectral changes?
I mean, it's a good question. So here we have the problem of the, as you said, the activity, the EMG, okay, activity and the EEG, and the certain frequency ranges perfectly overlap, So one might say that, well, what you see in CZ electrode is actually not EEG, but a muscular activation that actually looks like EEG because they overlap, which is true for certain frequencies. But what we can do is actually maybe perform some kind of blind source separation, like ICA and all the variants of the ACA. And there are papers, for example, that help us to cluster and to identify based on machine learning what the signals comes from the the muscular activation and what sources, what signals comes from brain cortical activations, and then we can try to separate them. Of course, this is not 100%, and we also have to be careful, okay, to doing that.
Yeah, thank you. And I think this is a good note to end up with. We have to always keep an open eye on improvements and also be proud on the progress that we already had implemented in such analysis. Thank you very much again for being here with us, for your time and your input to this workshop.
Thank you.
So after a short break, we will come back for the closing of the workshop and see you in a couple of minutes.